Okay, so this is the first video for the presentation corresponding with part 1, chapter 3. And one of the major questions that there are in genetics is how many genes are there in our genome? So that's one of the questions we're going to deal with today and try and make it happen. Okay. So why is it so important to know how many genes there are? First of all, for a very simple reason. If you know there's 100 and you found 80, you know how much you need to look for, right? That kind of makes sense. But it also has a very important part in understanding gene expression. So the genome contains all the genes needed for an organism, but not all the genes can be identified by genome alone. The transcriptome of RNA is actually expressed, but might contain more mRNAs than actual genes at the cellular level, meaning that there are more genes to be found in different levels of cell expression. When we're talking about the proteome, it is all of the proteins in an organism. There is a good but not perfect linear relationship between the proteome and the number of mRNAs. Remember, it's not perfect. And we talked about mechanisms that allow us to have more proteins than actual genes. Proteins act by themselves or within protein assemblies and complexes. So it's really important to the reason we want to know the number of the genes is to find answers to all those points that I've just mentioned. So theoretically, the key to finding out the number of genes is a genome that has only an open reading frame, right? Or operates with genes that use only ORFs. That would be perfect, right? Because that's the easiest case in deciphering a gene. But since interrupted genes have many ORFs, this task becomes extremely difficult. If we had one ORF and all the genes were uninterrupted, we wouldn't have a problem, would we? In addition, we don't know all the functions of proteins. So they're not known, they're not completely understood, or even expressed. There might be functions that are not expressed at a certain point. So it leaves room to imagine the genome's potential. But the assumption that we have is that any open reading frame will be expressed. That's an assumption. Is it completely true? Once again, it's an assumption, so it doesn't mean that it's definite. So the number of genes is actually the number of genes expressed plus those that are not expressed. If we were to find all the mRNAs that exist, this would lead to the total number of genes, because we know that the mRNA is a phase after gene transcription, right? So that would be what we call discovery via transcriptome, or by proteins, discovery by proteome. So either mRNAs or peptides, meaning proteins, would have to completely be found to answer the question of how many genes actually exist. So now we have a few questions that we can ask at this point. How many genes are expressed? We don't know the exact number. What variation of expression exists? Because we talked about patterns and modules of expression, right? How many genes that are expressed are unique to us? Are all genes essential? Not necessarily. Is a deleted gene always unessential? So if you remember, we assumed that if a gene is essential and it's deleted, it will lead to death. Does the lack of a gene affect us momentarily only? Is it something, or is it going to affect us for life? So those are interesting questions. Now you are referred to a movie I hope you enjoy. So in order to define a genome, we need to map it. And mapping the genome can be done at a few levels. The first level is to use the distances between mutations as a presentation of the recombination frequencies, meaning that mutations are mistakes, they are presenting recombination frequencies, and thus they are a certain marker or value that can be used to understand and map the genome. Of course, this is not perfect because mutations are not always present in the phenotype, right? So we know that if a mutation is not expressed, we'll see it in the genotype but not in the phenotype. So that might be a problem. It would be easier if all the mutations theoretically were present in the phenotype, meaning something we can actually see, observe. We can use recombination between genomic DNA. It can also serve for mapping. And what we would be using there is the idea of the sequence being susceptible to cleavage by restriction enzymes, right? So how open is the sequence, or how rare or not or common is cleavage by restriction enzymes. But this is not affected by the occurrence of mutations. It is still dependent upon recombination. The third option would be to map physically DNA according to cleaved fragments, but it does not always represent sites of interest, meaning that what if there's a site that I'm interested but it's not cleaved? So it also wouldn't represent the links between mutations and restriction sites. The major problem with this method would be that point mutations would be very hard to it will be very hard to detect them, right? We can sequence DNA. Meaning trying to find simply the sequence as one whole sequence, and that theoretically would produce the ultimate map, right? And then through transcription and the proteome, we could know the function and natural selection of mutations. Obviously, since it has not been completely done and we don't know everything, that's not as easy as it sounds, but in theory it can work. So when we're talking about mutations, a mutation's nature is determined by comparison to the wild type and to the mutation itself, meaning what kind of mutation is this, what is the outcome of the mutation, is it an insertion, is it deleterious, is it going to give us an advantage, is it going to give us a disadvantage, is it going to produce a disease, maybe it has no effect, right? So by doing that comparison, that will define the relationship between the genetic and physical maps, meaning what's going on at the level of instructions and information and what is actually going on at the physical level in the body. We have overlapping segments, right? Segments. So that assures that we have continuance of the genome as a key principle of accurate mapping. So when we have overlapping segments, it makes it easier to conduct mapping. So here's the thought. We have genes and we have multiple alleles, right, that exist. So how does that refer to the idea that we're using the term wild type? It becomes a bit problematic. So if we were to stay at that level, we would have a problem with explaining the variety and diversity in the population. So in comes a new term called polymorphism, and polymorphism is defined as more than one allele at the same locus, at the same site, and that gives us that resolves the problem. So we're talking about polymorphism as people having, or organisms having, more than one allele at the same site of a gene for a certain trait. Okay, so when we ask why are people different from each other at a certain aspect, we're talking about polymorphism. If we are identical within a certain population, completely identical, then there's no polymorphism. Let's say that we are examining a certain population. Let's take all the residents of a certain city, okay? And we see that a certain site has exactly the same multiple alleles in the population. So we will say that that is a polymorphic site. And in order for it to be called a polymorphic site, above 1% of the population should have it. Okay, so that's the cutting point. While the mutation map of a person, meaning simply mapping the mutations of a person, right, may lead to one conclusion, that polymorphism exists amongst mutant alleles. Moreover, they might be completely polymorphic. What does that mean? It means that if we map mutations, we might find out that alleles are basically polymorphic because of those mutations. When we look at the phenotypes that exist for different traits, it actually makes it hard to prove that wild types may be polymorphic, since not all sequence differences are apparent in the phenotype. So think about it for a second. There's a difference. It's not expressed in the phenotype, right? So you can't actually see a difference. It's in the sequence. So how do you show through a phenotype that wild types can actually be polymorphic? That would be problematic. So the way to look at polymorphism is at the level of the gene. That's probably the bottom line until now. You want to prove polymorphism, you go to the gene, because it's not necessarily going to be evident in the in the phenotypes or phenotype level. So we used in a video in chapter 2 the term SNAP, single nucleotide polymorphism. This is a very important term that I want you to remember, and we'll talk about it shortly. So you're referred to a movie. Alright, so the SNP, SNP, is a change of one nucleotide only between alleles. So let's say we take two alleles, right, that are going to lead to two different phenotypes, and we compare them. We find a difference of one nucleotide between them. In average, every about 1,300 base pairs, a SNP occurs. They occur uniquely, meaning that makes that person unique. It's like a fingerprint. So theoretically, once it will be easy to take someone's genome and just map it completely, we will find a unique pattern of SNPs. And that means that that's your fingerprint. There's no two that are identical. They might be similar, but not identical. So it's your genetic fingerprint, so to speak. These SNPs are also called variants. They're called variants because they distinguish between people, right? So if we can map all the SNPs in the population, that would be the best way, or one of the leading ways, to learn about the human variation. So restriction maps are actually tools of SNPs comparison. Through a change in the cleavage pattern, we also know the pattern of fragmentation. So once again, a SNP is a unique character to you. It happens in certain places only in your body. No one in the world is supposed to be identical, right? Theoretically. So if we're talking about cleavage and we're talking about restriction sites, those will be unique to you because you have unique SNPs. Does that make sense? I hope it does. So restriction maps can show polymorphisms regardless of gene function or phenotype. Okay? Most of the polymorphisms are not going to affect the phenotype, and the majority of them are actually located between genes. Okay, we're going to stop here and continue in the next video.